Welcome everyone, and thank you for joining us for today's webinar titled Automated Analysis of Behavioral Data, Examples from Fear Conditioning Studies. My name is Andy Henton from Inside Scientific, and I will be your host for today's event. Today's session is the fourth installment in our Animal Behavioral Neuroscience webinar series. As of today, nearly 600 scientists from around the world have registered to participate in this web series, and I would like to extend a warm welcome to those of you who are online with us today. Thank you for joining us. Our session is sponsored by Med Associates and is focused on the topic of automated scoring and analysis in fear conditioning research using laboratory rodents. Specifically, our talk will focus on the key principles behind fear conditioning paradigms, correct protocol design, and the importance of both methods and equipment validation.
Okay, thanks, Andy. And it's a pleasure to be here. I'm glad to have all of you here. Thank you all for joining us on our webinar today. Today we're going to talk about automated analysis of behavioral data, specifically using fear conditioning studies as an example. So in today's webinar, we're going to look at starting off the basic concepts in fear conditioning and the components that make up a video-based fear conditioning or system for detecting freezing behavior system. Once we've talked about the basic concepts, we're going to then talk about the principles behind automated motion analysis and proper detection of freezing. So how does an automated system detect motion and determine freezing behavior? And specifically here, we're also going to hit on one of the key points of today's webinar, and that's the importance of validating settings. So we're going to spend some time talking through how you can approach validating an automated system to make sure that the automated system is scoring behavior the way that a trained human observer would, for instance. And finally, we're going to end by looking at a few applied examples of fear conditioning experimental study designs. So let's first talk about some of the concepts in a fear conditioning study. This slide illustrates the different phases of a fear conditioning study. First of all, an animal, rats and mice are very commonly used. The animal would be placed in a training environment, and this is a conditioning chamber that allows for the presentation of various stimuli, such as audible stimuli or sounds and tones. There's usually provisions for delivering visible stimuli, such as lights. And then there's also the floor that the animal is on has grid rods that can be connected to aversive stimulation. And then in the case of fear conditioning, it's a form of classical conditioning or Pavlovian conditioning where you have a normal neutral stimulus, such as a tone, being paired with an aversive stimulus, such as a foot shock. Now, in the training phase, the animals usually explore their environment. They're inquisitive. They're investigating this environment. Then, once the tone and the shock pairings are done, they begin to exhibit freezing behavior, which is just shown by the animal crouching down here in the corner of the cage. And so, in an automated system, it should be designed to allow you to present stimuli, audible, visible stimuli, present aversive stimulation, and also detect when the animal is moving or not. Once the conditioning is done, typically an animal would be placed into a novel context, a novel environment. That's symbolized here with the darker environment here. So under training, the training environment can be brightly lit. The contextual shift can be done in a darkened environment or vice versa. And different inserts can be placed into the cage to allow you to present a different environment to the animal. Oftentimes different odors are also placed. So one odor would be used for training, a different odor for testing, for example. And in this novel context, the animal again exhibits inquisitive exploratory behavior, as seen here with the mouse kind of sniffing out his surroundings. But then when the tone is presented that was used during training, the animal exhibits the freezing reaction. And again, an automated system should be able to detect motion in these different environmental contexts and determine when the animal is moving or when the animal is freezing. Today we're going to talk specifically about the MetaSociates video freeze system as an example of an automated behavioral analysis system. And this screenshot shows an image from the video freeze software where you see a live video feed of the conditioning experiment in progress. And in this training phase, again, when the animals are first placed in the environment, they're typically exhibiting high levels of activity. This is exploratory behavior. And we use a motion index in order to determine if the animal is moving or freezing, and we'll talk more about the motion index a little bit later. But with the motion index, you can see here the green line, high motion index symbolizes high levels of activity. When the motion index is low, that would indicate the animal would potentially be freezing. So here is a motion index from a training trial. So the first time the mouse is in the chamber, he's moving around, exploring the cage quite a bit. And then down here on the bottom, we see the experimental timeline. So we see in this example, there was a period of several minutes where nothing was done. The animal is just, it's a baseline condition. The animal is allowed to explore the chamber. And then a sound is presented. Usually this sound would be 30 seconds in duration. And at the end of that sound, we have a 2-second shock presentation. So this is the first time the animal heard the sound, and then at the end of the sound, a foot shock is delivered. You see the motion index jumps up very large when the shock is displayed, and that's the animal jumping in response to the foot shock. And then in this period between the first stimulation and the second stimulation, we see a lower level of overall activity, and then the second foot shock, you can see the animal jump up here, and by that second trial, the animal is freezing quite a bit, so we see very low levels of activity. And then the third tone shock pairing is here at the end, and we see the animal jump again, and then low levels of activity, high levels of freezing. Okay, so that would be the training phase. Contextual cues are very important, as we talked about odor, illumination levels, the cage geometry, are all important contextual cues that the animal begins to associate with this aversive event during training. This is a video of a training session from a C57 Black 6 mouse, and I'm going to play this video just to illustrate the what happens. You have the initial high levels of exploratory behavior, and then you won't be able to hear the sound, but I'll tell you when the sound is coming on, and then you'll also be able to see the mouse jump when the foot shock comes on. So I'll call out those events when the tone comes on. So I'm going to play this video just to show you what the animal's behavior is like during the training phase of the fear conditioning experiment. Okay, so we have the animal moving around, sniffing, investigating. This is all typical exploratory behavior that the animal displays in a novel environment. Now the tone has come on, and this is the first time the animal has heard the tone, so he pays no attention to it. Now the foot shock is on, and you can see the animal jumping. The animal was also vocalizing at that same time, but now the animal's activity is greatly suppressed, and we start to see some freezing episodes. Now the video is stopped. Okay, so that's a typical training session in a C57 Black 6 mouse. Sometime after training, usually this would be done the day following training, would be done what's called the context test. So in the context test, the animal is returned to the training environment and simply left there for a period of time. Maybe 5 minutes is a typical duration for a context test. So there's no stimuli delivered. We see on the timeline there's no stimuli being applied here. The animal is simply placed in the training context and his behavior is observed. And what you see is that the animals will usually exhibit freezing behavior right away when they're placed in this training context. And this is interpreted as fear to the contextual cues in the training environment. So the odor that was present, the light level that was present, any patterns or shapes that are present in the training context. The grid floor is another important contextual cue. The animal recalls all of those cues and remembers that the day before you didn't like being shocked in that chamber. So the manifestation of that is the freezing response. And on the motion index, we see very low levels of activity. If we look back quickly here to the first day, the training, we see high levels of activity initially in this environment. Now, after the animal's been fear conditioned, the activity is greatly suppressed, lots of freezing. And so let's look at a video of a context test. So this is a video of the same mouse that we just saw the day after training. He's placed back in the same context. And I'll play this video here for you. And the video is playing now. You can see the animal do a few head twitches, but for the most part, the animal is freezing. Okay, and the video is still playing. Now the video just stopped, but you see the animal is pretty much staying in the one spot, exhibiting a lot of freezing behavior, and that's a typical manifestation of that happens with fear conditioning. So another important phase of a fear conditioning study is the tone test or the cued freezing test. In the tone test or cued freezing test, this is also done some period of time after training, typically 1 or 2 days, or depending on the experimental goals, that could be even longer. The animal is placed into a new environmental context. And you can see here the context is altered by inserting an A-frame into the chamber, also putting a cover over the grid rods. In this case, the visible lights are also turned off. So training was done in a brightly lit chamber. The tone test is done in a dark chamber. So from the animal's perspective, this is a dark, very small enclosure. So what happens in this novel environment is the animal initially shows high levels of exploratory behavior. And we look at the motion index, we see the animal is moving around quite a bit here, investigating his surroundings, learning about this new environment he's in. And then after a baseline period, the tone or the conditioned stimulus that was used during training is presented to the animal, and that brings forth the freezing response. And in this case, we've done 3 presentations of the conditioned stimulus, and we see freezing in response to each presentation of the tone. So let's look at a video of a tone test here. Again, this is the same mouse that we've been following, placed into a novel context, the tone test context, and we have the A-frame and the smooth floor over the grid rods. And I'll play this video for you, and you see that the animal's moving, exploring the environment, sniffing, getting to know the surroundings. And now the tone has just come on, and you see the animal goes to the center of the cage and starts to freeze. Okay. And now the tone is off. The animal moves a little bit and starts freezing again. So this is typical reaction in a mouse that's in a fear conditioning study. Okay. So an automated system should be able to track behavior in any of these contexts, whether it's a training context or a test context. The automated system should be able to score behavior regardless of environmental context. This slide here shows the equipment that is needed to perform such a study in fear conditioning. These are the components of a video-based fear conditioning system. We have a sound attenuating cubicle that's going to help isolate the conditioning chamber, prevent it from being interfered by noises in the lab or other events going on in the lab. There's a video camera, a light source. We'll talk about the camera and the light here in a few more moments. The conditioning chamber, which allows for the presentation of conditioned stimulus such as a tone or a visible stimulus light, and also has the grid rods for delivering foot shock. One important consideration with behavioral studies is to use acoustic foam lining. This is not only for fear conditioning, but any behavioral experiment. You can improve isolation by lining a cubicle with sound attenuation foam. And the importance of that is, for example, in fear conditioning, you're going to have the tones being played in the test chamber. when the animal is being shocked, the animal will vocalize. You want to prevent those noises from interfering with conditioning cubicles that are adjacent to this cubicle. So you want each cubicle to be not influenced by neighboring cubicles. So the sound attenuation lining can help you achieve that improved sound isolation from one chamber to the next. And that's a good principle for any behavioral studies, whether it's fear conditioning or any other kind of behavioral. So now with a camera in a video-based system for fear conditioning, the camera is a very important component in the system, as is the illumination. And since an automated fear conditioning system must measure animal movement, a low noise camera is essential. So in an automated system, the goal is to have the computer determine if the animal is moving or freezing. And if the camera being used has a lot of inherent noise or is a low resolution camera, it's going to be much harder to determine animal movement from just background video noise. So the use of a low noise camera is really essential. Illumination is just as important. In fear conditioning, visual cues are often used. So you often have a stimulus light that is used to as a conditioned stimulus, or perhaps the visible light levels are manipulated as a part of the environmental context. You might have bright lights in one context and very dim or no lights in another context. Those manipulations to visible light should not interfere with the way the automated system works. In the MedAssociates video freeze system, we actually achieve that by using near-infrared lights, and that was an important innovation that we brought forth when we introduced our video freeze system. We actually use a filter that blocks visible light, so you can turn visible lights up maximal or any one of 5 different intensities, or you can flash lights on and off as part of the training session. And that does not interfere with the video signal because we block visible light from ever entering the camera. The camera only receives the infrared light, and so that remains constant throughout the experiment. So we always have a really well-lit image, well-illuminated image from the camera's perspective. And that's an important consideration. Some other video-based systems have the confound that if you turn cue lights on and off, the flashing of a light can produce a stimulus artifact and be interpreted as motion by the system. So you have to be aware of that with any automated system. Also, the contextual inserts that are used, it's necessary to alter the context of the testing environment in fear conditioning. So the automated system should be able to score behavior similarly in multiple contexts. This is showing a conditioning chamber with what we call a curved wall insert. So it changes the geometry of the cage from a normal box type of enclosure to a curved wall, and that can provide a nice distinct environment to do a contextual discrimination. And the automated system should be able to score behavior regardless of the configuration of the chamber. Here's another environmental manipulation showing the A-frame and the smooth cover over the grid rods. In this case, any kind of manipulations that are made to the conditioning chamber should not interfere with the illumination or the ability of the system to detect and score movement. Regarding the grid floors, this is an important element of contextual cues. The grid rods themselves are a strong contextual cue. If you take an animal that's been fear conditioned with foot shock exposure on grid rods, you can put them into a greatly different environment, but if they're still on grid rods, just the grid rods themselves will present a strong cue to the animal, and they'll show high freezing levels. So that to minimize kind of any carryover of freezing to a new environment, it's often advisable to use a drastically different floor, and that can be achieved by putting a smooth cover over grid rods or using grid rods of different patterns where you have like alternating large and small grid rods, or maybe they're staggered in a zigzag pattern. Those are all things to consider as far as changing the environmental context. Okay, so now that we've looked at the components in a fear conditioning system, how do we measure freezing behavior in an automated fashion? How does the system work? Well, a video-based system that measures freezing behavior must have a way of detecting when an animal is moving or not moving. That's inherent in the test. When the animal is not moving, or when there is no animal present at all, the system should score 100% freezing. And that's kind of may seem silly, but actually that's an important concept. If you're initially testing a new system for freezing analysis, and one thing it would be advisable to do initially is just take a recording with no animal present and see what the system scores for freezing. In that case, if there's no animal present, you should get a score of 100% freezing, because there's, by definition, no animal, no movement. That should be 100% freezing. If you get some level of freezing less than 100%, that's an indication that there's some noise in the system that could be interpreted as movement and may reduce the ability of the system to detect freezing. So that's kind of a good test with a new system, is just take a recording with no animal and see what the score is. On that same point, subtle movements such as grooming and sniffing should be differentiated from freezing. So if the animal is making very subtle movements, grooming, sniffing, investigating the cracks in the cage, those would not be considered freezing by a human observer, so the system should not score those as freezing. Okay. There's a number of different approaches that can be used for tracking animal movement and scoring freezing behavior. It can be done with photo beams, video-based, or load cell-based. Those are some of the more common ones that are being used today. And regardless of what type of system is used, the goal is to determine if the animal is moving or not moving. And with that consideration, the tracking of animal movement should not be highly influenced by experimental variables. So whether there's different contextual inserts used or different light levels, none of those manipulations should greatly influence the way the system scores freezing. In the case of our Metassociates video freeze system, we used a motion index for determining if the animal is moving or not. And the motion index captures animal movements to distinguish slight movements from freezing. And this works regardless of coat color, animal size, environmental context, and visual cues. So we use a proprietary tracking algorithm that is constantly analyzing the video feed, and it's comparing the video signal in real time against a reference signal of background video noise. And that's how we're able to determine if the animal is moving or not moving. We're continuously matching the video signal against a noise reference. And so by doing that approach, we're able to get very high resolution for determining small movements and differentiating those from freezing. And this just shows the motion index here. As we talked about earlier, when the motion index is high, the animal is moving, and when it drops down low, the animal may be freezing. Okay. And the way we determine if the animal is freezing, there's two parameters that the user has the ability to set. There's the motion threshold, and then there's also the minimum freeze duration. And on this slide, I'm just illustrating idealized motion index here over time. So the motion threshold is an arbitrary limit that the user sets to determine below which the animal may be freezing. If the motion index drops below the threshold, the animal may be freezing. The other criteria that has to be met to be counted as freezing is that the motion index must be below threshold for the minimum freeze duration. So there's a temporal element, a time element to the analysis, as well as the amplitude criteria. So if the motion drops below threshold and remains there for the minimum freeze duration, the animal is considered freezing. If the motion index exceeds the threshold at any point, the animal would be considered moving. Okay. So the dependent measures that come out of the system are, you get a % freeze score. That's the time freezing divided by the total session time. You get a count for the number of freeze episodes, the number of times the animal freezing, and the freeze duration, the total amount of time the animal spent immobile or freezing. So now that we've talked about how the system scores freezing, let's talk about how you validate settings. And that's an important concept for any automated behavioral system. And that is taking computer scores or the automated system scores and compare those to trained human observer or hand scoring. That's an approach that should be done regardless of what type of experiment you're doing. It's always best practice to validate the automated system against a reference, a known reference. It's a key step in data collection, validating the system. Validation is going to ensure that the system being used is analyzing the behavior in question in a reliable manner that is consistent with traditional or accepted methods. So it's really advisable to validate any automated behavioral system. The way this, Dr. Anagnostaras from UCSD, we collaborated with him a few years ago and published a paper in Frontiers in Behavioral Neuroscience, and if you that paper provides the framework for how you can approach validating an automated behavioral analysis system for fear conditioning. And some of the premises behind this validation approach is that you need to have a way of determining if the animal is freezing, and so what is the definition of freezing? Freezing is often defined as the suppression of all movement except that required for respiration. And there's some, this kind of definition of freezing is well established in the literature, and here's some suggested references and resources that can be looked at for further information. Traditionally, the way freezing behavior would be manually scored would be that you'd have a trained observer who is looking at a video monitor and they're taking periodic observations. For example, every 8 seconds they would look up at the video and make an instantaneous judgment. Is the animal freezing, yes or no? And again, what's the definition of freezing? Lack of all movement except respiration. So is the animal moving or is he freezing, yes or no? And that instantaneous judgment is made at periodic intervals throughout the entire session. And then you tally up your observations. You take the number of freezing observations divided by the total number of observations, multiply by 100, and that gives you your % freezing score. An alternative method that's used in some labs is a continuous method, where the human observer is continuously watching the video and they're starting and stopping a stopwatch every time the animal starts and stops freezing. In that case, the time spent freezing is divided by the total time multiplied by 100 to give you your % freezing score. And those methods, this time sampling method versus the continuous sampling method, have both been used extensively in the literature and are both well accepted and validated. So to validate an automated system, what you want to do is you want to take observers trained in either of these methods to score your data set and then compare that to what the computer is giving for scores. And in the Anagnostaras et al. Paper in 2010, we provide that an outline for how that can be done in a video fear conditioning system. And this approach can be used as a reference for general validation of any automated behavioral analysis system. So in the paper with Dr. Anagnostaras, we made some assumptions and set some premises that we were trying to follow, and those are shown here. So since freezing is defined as the absence of movement, the system should measure movement in some way. We have to be measuring movement and determining if the animal is moving or not moving. If near zero movement levels would then be equated with freezing. Second, the system should detect small movements, such as grooming, and not count them as freezing. If there's no animal present, the system should score 100% freezing. In other words, the system should reject any video noise. If noise is counted as movement, that's going to give you, that's going to diminish your ability to detect freezing. Okay. The next point was that signals from small movements such as grooming should be well above video noise. If the animal is grooming, that signal should be well above video noise in order to differentiate it from video noise. And the detection should be fast. You shouldn't have to wait hours and hours for a cohort of data to be analyzed. It should happen fast, ideally in real time. And then lastly, and most importantly, the scores generated by the computer should correlate very well with scores obtained from multiple trained human observers. And what do we mean by very well? That's illustrated here in this best fit line. The correlation coefficient between human scores and computer-generated scores should be near 1. Should have a very high correlation coefficient. The fit should have a near 0 y-intercept and a slope of approximately 1. If those criteria are met, then that means the computer scores and the hand scores are matching up exactly 1 for 1. And so we have a perfect straight line with a slope of 1 that passes through 0 on the y-intercept. So that's the ideal case where the computer scores freezing exactly the same as trained human observers. 10% freezing is 10% freezing, 20% is 20%, and so on. Okay, so that's the ideal case that we're after here. Here's some examples of the validation that was done with our video freeze system in mice. And this is from the Anagnostaros et al. Paper 2010. We have correlations done between the video freeze software on the x-axis and hand scores, and I think this was from 3 trained observers, the average of 3 observers here on the x-axis. And we see under visible light conditions, we see a very good correlation between the automated scores and the hand scores. The fit values are shown here in this little inset. So the slope is near 1, 0.968. The y-intercept is very close to 0, negative 0.4. And we have the correlation coefficient 0.968 is very high. So this indicates a really good fit between the computer and the hand scores. We achieved a very similar fit regardless of lighting conditions. So when the white lights were on or when only the near-infrared lights were used, we see very good correlation. So the automated system is not influenced by changes in visible light. And then the Bland-Altman plot is just a way to correlate the reliability of the computer and hand scores in terms of how much variance there is between the two. Now, a little bit more details on the correlation and how to validate correlation fits between automated scored data and hand scored data. In the case of video freeze, as I mentioned earlier, there are 2 parameters that you can vary. There's the motion index threshold and the minimum freeze duration. So we looked at correlations by varying those 2 parameters. On the x-axis here, we were varying the minimum freeze duration in terms of number of video frames, and on the each symbol type is a different motion index threshold. So what we were looking for in our initial validation was finding the set of parameters, the set of thresholds and minimum freeze durations that gave us the highest correlation coefficient, the y-intercept closest to 0, and the slope closest to 1. So again, we went through and tested a series of minimum freeze durations at different motion index thresholds, and we correlated the human scores and the computer scores by varying those parameters. And in the end, we were able to determine the best set of parameters that gave us the highest correlation coefficient, the y-intercept closest to 0, and a slope closest to 1. And that should be the goal of any automated system, is you'd want to, whatever the parameters that you can vary are, you'd want to examine a series of parameters and find those parameters that most closely match the way the trained observers would score the behavior. Now, I want to look at some possible scoring outcomes here. These are some possible outcomes you can see when you're attempting to validate an automated system. The first example that we look at here is a system in which the automated system overestimates freezing at low levels of movement. Okay, and in the graph here, I'm showing the ideal best fit of a perfect line here in the black symbols again. So that's our, ideally, the system would score freezing like this, a perfect straight line passing through 0 on the y-intercept with a slope of 1. It's possible that the computer scores would overestimate freezing at low levels of activity, as shown by the red symbols here. So at the case of low levels of freezing, 10% freezing according to the human is being picked up as 20 or 25%. 30% freezing according to the human is being scored as 60% or even 70%. So the automated system is giving us higher scores than what the trained observers are scoring. So that's overestimation of freezing. Okay, so that's one possible outcome. And if we do a fit of that data, we would see that the slope may or may not be near 1. You may have a good slope. You may have a relatively low correlation coefficient, but very importantly, the y-intercept is going to be a positive, a significant positive value. It's going to be greater than 0. So we see the best fit line to this data is passing through 20% on the y-axis. So we get a y-intercept greatly above 0. That's a red flag that the system is overestimating freezing if you have a positive y-intercept. So that's something to watch out for. Number 2, the second case, before we go on to the next case, what are some possible causes that can lead to overestimation of freezing? Well, in the case of video freeze, again, the 2 parameters that can vary are the motion index threshold and the minimum freeze duration. So overestimation of freezing could be caused by the motion index threshold being set too high. So we're scoring too much behavior as freezing. And the same thing would happen if the minimum freeze duration is too short. If we have too short of a time be considered freezing, then we're going to be counting more low-level activity as freezing than what is actually happening. Okay, so either of those cases would cause overestimation to occur. The next case would be the automated system underestimates freezing at low and even mid levels of movement. So again, we have our perfect fit here, our perfect agreement between the computer scores and the hand scores in the black line. The green symbols are showing a system that underestimates freezing. So say the human observers are scoring 20% freezing behavior, the computer is only picking up less than 5%. 40% behavior freezing, but according to the human, is coming in at 15% according to the automated system. If we do a best fit line of that data, it might look something like this. Here, the green dots. And what you would see, the slope may or may not be near 1, and the correlation coefficient may be low, but most importantly, the y-intercept is going to be less than 0. This line is going to pass through the y-intercept at some negative value, significant negative value. And that's a y-intercept less than 0 is a red flag that your automated system is underestimating behavior. So what can cause this? Again, in the video freeze, we have the motion index threshold and the minimum freeze duration. So if the motion index threshold is set too low or the minimum freeze duration is too long, both of those scenarios would lead to the system underestimating the freezing behavior. If the motion index is too low, we might have the animal freezing, but the motion is above threshold, so it's not going to be counted as freezing. Or if the minimum freeze duration is too long, the animal may be freezing, but may be doing so in shorter bouts, and so we're never reaching that temporal criteria to be counted as freezing. So either of those situations would result in an underestimation of behavior. Let's now look at a really significant challenge with detection of movement, and that's measuring subtle and brief movements. Okay, this is the most significant challenge for any automated system that detects freezing. How to distinguish subtle movements from actual freezing. Okay, I've got a few examples to show you. This is a video. I'll play the video here in a moment. I just want to walk you through it here first. If we look at the motion index threshold, the blue cursors here are bracketing this region of time. It's only a few seconds long. And this is the part of the video that we're going to be looking at. I'm going to loop it a few times for you. And what I want to show you is I'll call out in the video, you'll notice the spike in the motion index. That's going to correspond to the animal, the rat over here, making a very brief head bob, a very brief adjustment in the angle of his head. and that would be scored by a human as breaking a freeze episode. When the animal is moving his head, that's not freezing, okay? And so we want to make sure our system is not counting something like that as freezing. So let me show the video to you now. Again, we'll loop through it a couple times. So, okay, the animal's freezing, and there's that really brief head bob, and now he's freezing again. Again, one more time, freezing, there's the head bob, and we're back to freezing, okay? So by adjusting the motion index threshold, we can alter whether or not that slight event gets counted. So those types of slight movements are very easy to miss in an automated system. Okay, if we now take this motion index threshold, this part of the motion index threshold right here, and zoom in on it, that's what we're doing in this graph here. So I've just taken that brief segment of the motion index, and I've expanded it so we can see it very closely. We see we're only looking at a few seconds here, from 687 seconds in the session to 691. I've drawn a red line at the motion index threshold, and I've drawn a red box to indicate the minimum freeze duration. And so what we see with that very subtle head bob, the motion index went above the threshold and then came back down again. So it's only during the times when the motion index is below the threshold for this entire window here for the minimum freeze duration, that's when the animal is considered freezing. And being able to detect those types of subtle movements is the most significant challenge for any automated system that scores freezing behavior. Okay, now let's look at one more example. This is a little bit different. We have an albino rat this time, and the albino rats, this is a Wistar, and it's a female Wistar, and she's doing a lot of very brief freezing episodes. And I have this region of the video here from this blue cursor, or it actually looks more white, to this cursor here. This is the region of the that we're going to be playing in a video loop here. And what's going on here is we have one episode of freezing right here that I'm circling. That's the one freezing episode. And then the rest of this is movement. You'll see the animal sniffing and investigating the cage. So let's watch the video. Okay, freezing. Now the animal's moving around, sniffing the cage. Now she comes to the front of the cage and is sniffing this little crack between the door and the cage floor. And we'll loop through that again. So the animal's freezing, and now she's moving. And when we're looking at these subtle movements, these subtle sniffing movements, that can easily be missed and counted as freezing. But with the appropriate motion index threshold and minimum freeze duration, we can distinguish those movements from actual freezing movements. So let's zoom in on the motion index and look at that a little closer. So this is that same region of the file we just looked at. Now we're just zooming in on the motion index threshold, and we see the motion index threshold in the red line and the minimum freeze duration with the red boxes. I've highlighted two segments here. This first segment is the one where she's actually freezing, and then all the rest, the animal is moving around and sniffing. Down here, this is a really tricky part because if you just look at the motion index, it looked like she might have been freezing, but when you look at the video, she was actually sniffing the front of the cage. And the criteria that's invalidated here is the minimum freeze duration. So because she was not below threshold for the entire minimum freeze duration, she's not counted as freezing. Okay, so if we had our motion index, our minimum freeze duration too short, we might count this as two different freezing episodes right here. But by having a long enough minimum freeze duration, we can distinguish that from actual freezing. Okay, so there is a risk of overestimating freezing if the minimum freeze duration is too short. Okay, and now finally, let's look at a few examples of experimental study designs. We'll look at context discrimination and shock reactivity. So these are some applied examples of fear conditioning. With context discrimination in rats, We're actually in the process of doing a study in our lab right now validating the video freeze system with rats. As I mentioned, the Nagnastaras et al. Paper came out in 2010 in Frontiers of Behavioral Neuroscience, validating the system with mice. As we speak, we're actually finishing up the study in our lab to do the same thing with rats, to validate the system with rats. And this is an example of some of the experiments we've been doing. So we have been training the animals in one context, and testing them in two additional contexts. So we have a training context we call A, and then we have two test contexts, B and C. Training is done with bright lights on. Testing contexts are both done under with visible lights off. And we're using two different strains of rats. We're using Wistar and Long Evans rats, and we've got 8 rats in each group. And we're also looking at different shock intensities. For today, I'm just pulling out examples from rats that were trained with 1 milliamp of shock for the aversive condition. And under baseline conditions in the training context, we have very low freezing. Again, the animals are exploring and they're exploring their environment, so they show very little freezing under baseline conditions in the training context. However, immediately after training, in their training context, they show very significant freezing. So the learning happens very fast. On subsequent days, we've placed the animals into the 2 additional test contexts, context B and C, and we see very low baseline freezing scores. So this is, we don't see a lot of carryover from one context to another. That's a good sign to look for in fear conditioning. With acute freezing test, we see high freezing in both strains of rats in all contexts. So when we play the conditioning tone in context A, the training context, we see significant freezing in both strains of rats, and the same thing in the additional test context B and C. Now here's an example of a study design looking at context discrimination in mice, and this is just the experimental timeline where we had 5 tone shock pairings. On day 1, the training occurred in context A. On day 2, we did a test in the training context. So we just put them back in the training context. On day 3, we put them into a new context, context B. And this shows the training data, the so-called learning curve. So under baseline conditions, again, the mice show very low freezing, essentially 0 freezing prior to fear conditioning. During the first tone, they're still not freezing. This is the first time they've heard the tone, so they really pay no heed to it. However, the next time they hear a tone, they show high freezing or increased freezing, and freezing response increases with successive tone presentations. And then in the period at the end of the training session, they're showing high levels of freezing, about 25% on average. Now, the next day, we put them back into the training context and do a tone test. And under baseline conditions, they're already freezing 20% of the time, or 15 to 20% of the time, which is carryover from the training context. Okay, so they remember being shocked in this environment. And then we do a tone test where we present the training condition stimulus, and the freezing increases even more. The next day, we put them into a new environmental context, context B. They show low baseline freezing. Again, that's a good sign. They're showing inquisitive behavior in this new environment. However, in response to the tone presentation, the condition stimulus, they show high freezing. So they recall the tone and associate it with the shock that they received in the days prior. The last example I want to show you is an example of shock intensity in mice, and this is just to illustrate the concept that the amplitude of the aversive stimulus or the unconditioned stimulus can also impact the behavioral outcome. So in this experiment, we had 2 CD1 mice in 2 different groups. One group of mice we trained with 0.27 milliamps of foot shock, and another group we had 0.45 milliamps, and it just shows you freezing. The 0.27 milliamp group was freezing about 40% of the time, and the animals that were trained with a higher shock level of 0.45 milliamps are freezing 70 to 80 or even 90% of the time. So the amplitude of the aversive stimulus can also impact the behavioral outcome in fear conditioning studies. Okay, so just to summarize the talk, again, thinking back to the components and the equipment instrumentation, care should be taken to set up your behavioral assay in a way that limits interference from other lab activities. So using sound attenuation cubicles, using sound attenuation foam lining those cubicles, having a dedicated workspace if possible. All of those are really important considerations for any behavioral study, not only for conditioning. And importantly, when you're using an automated system for scoring complex behaviors such as freezing behavior, special attention should be paid to validating the settings that you're using. Make sure that the automated system measures behavior in a manner analogous to the way trained human observers would be scoring the data. And that's some of the best practices involved. Okay. I just want to end by thanking our team, our lab team here at Catamount R&D and Med Associates. This is Mary Beth Klinger. She has her PhD in neuroscience, anatomy and neurobiology from the University of Vermont. She's our lab director, and we have two research assistants, Mahalia McGill and Sierra Bruno, who work in our lab and are involved in our taking part in this rat fear conditioning study that I was describing to you. And we also have a summer student working with us this summer, Nakia Whiten, all a big help and a great help in our lab here at Med Associates and Catamount R&D. And again, I just want to thank you all for your attention today. I want to encourage you, if you haven't submitted questions, but you have one, submit them in the chat. Or if you have to, if you don't want to submit a chat question, we have a survey at the end. Please take time to fill out that survey and leave your questions there. We will respond to all your questions, even if we don't get them today. We will offline respond to all the questions that come in, and you will all get a detailed report of the question and answer session for reference materials. So again, thank you, and thanks to Andy and Martin at Insight Scientific for putting on the webinar today.
Great. That's wonderful, Gerry. Thank you very much. And you know, we have had a number of questions come in, so let's get right to it. As I mentioned through the chat window, we're a little bit over time, but we're going to have a brief Q&A anyways and answer some of these questions. So here we go. All right, first one is, do you need to have a certain set of parameters for each type of experiment you're doing, or put a different way, you know, how often should one validate the settings of an automated free scoring system?
Yeah, that's a really good question. Ideally, you should not have to have a different set of parameters across multiple experimental types. So, and validation should be done certainly at the beginning of a project. But once you've established settings for the system, you shouldn't have to change them. Now, I would make an exception to that if you're changing from looking at rats to mice, for example, or you're starting out with mice and then you move to rats. You would definitely need to address your validation because you might have different parameters for rats versus mice. But you shouldn't have, for example, in fear conditioning where you have a training context and maybe 2 or 3 different test contexts, you shouldn't need to have different settings for the different training contexts. You should have one set of settings that apply to all your experimental contexts. Again, that approach could vary based on the automated system that's being used, but that's in a robust automated system would be able to score the data accurately and reliably regardless of slight changes in parameters.
Perfect. Okay. Another question, how many human scores should someone plan to use to validate an automated behavioral data analysis? Is there kind of a rule of thumb or a best practice here, you know, on the number of human scores that would be involved in a validation study?
Yeah, it's another good question. I would say at least you'd want to have minimally 2 different human scores going in, and you'd also want to make sure that those that the scores that are used for establishing what you'd call ground truth in signal processing, your reference signal, you'd want to make sure that there's good correlation amongst your human observers. So if you have 2 people scoring behavior, you'd want to make sure there's good cross correlation between their scores before you average those scores to use in your validation. And if you're using more than 2, again, you'd want to make sure there's good cross correlation amongst all your scorers before they're used in establishing ground truth.
Okay, great. That's a good answer. Something specific to video freeze, a question has come up about the ability to reanalyze a video. And I think what we're trying to get at here is when we go back to our polling where we adjusted data, what capabilities does a user have to do that type of thing, and what are you really adjusting? So are you reanalyzing a video, or what's happening there?
Yeah. With the video freeze system, you can reanalyze data at will. So the video itself in real time is being analyzed. Raw video from the camera is being analyzed in real time, and a motion index is generated in real time. Now, when the video is saved to disk, it gets compressed. So when you're reanalyzing data, you're actually not reanalyzing the video. What you're reanalyzing is the motion index, and you can apply different parameters. You can apply different thresholds and different freeze durations to that motion index to determine which parameters best fit the data. So yeah, you can reanalyze the motion index data as many times as you want offline, and that really does help establish parameters that best fit to your human scores.
Perfect. Okay. And then what about the analysis settings? I mean, can you change that?
Yes, you can change the analysis settings at will. Once you've established your settings, you'd want to leave them constant. But yeah, in the validation phase, you can change the settings as much as you need to.
Okay, perfect. So here's a good question. Do you have any perspective on protocols where the desire to measure freezing is also combined with other animal behaviors, like grooming and rearing and subtle movements? So I guess one way to put this is, are there freeze systems capable of combining these measurements and analyzing maybe from a motion index or additional data that are coming in from a system so that you can do multiple collections simultaneously? Is that even possible? Are people doing that?
Yeah, there are some systems out there that are doing more complex behavioral recognition.
Mm-hmm.
The one really important thing to remember, though, is validation. If a system is designed for measuring grooming, for example, or some type of stereotypic behavior, maybe paw licking or rotational stereotypies,
Mm-hmm.
There's many claims made that systems will do this and automatically score these behaviors. However, if you look at validation, it's kind of all over. So some are like claiming 80% accuracy or 90%, but really the goal is to get right on with human scores. So I would just point out the need to still, if there's a claim made that a system is doing some behavioral recognition, that you really would want to pay heed to how the validation was done to reference what the automated system is scoring versus what's actually happening.
Okay, perfect. That's a great answer. We've got another one here. Mackenzie asks, how many pairings of CS and US would be sufficient for the training day? So how long in duration would the context test day be compared to the original testing day? Is there a best practice or a rule that needs to be applied here?
Well, I think a real kind of typical training session would have at least 3 presentations of the conditioned stimulus and the unconditioned stimulus, 3 tone shock pairings. And usually they're separated by 60 to 90 seconds. I would say 3 to 5 or 6 are very common, but you really probably want to have at least 3. But even a single trial elicits learning. So, I think you couldn't just say here's how many you should do, because that depends on the type of the experimental goals. But for basic fear conditioning, 3 is a good number to have for tone shock pairings.
Okay, perfect. All right, perhaps this will be our final question here. Jennifer asks, currently she is using 2 independent systems. She explains that day 1 training, day 2 is context in the same system as training, and then day 3 a tone test in that second system with inserts. The question though is it necessary to change to a new system in a different room for day 3 to really ensure that no context such as smell or the environment is accounted for during the tone test? So, I guess paraphrase is like, how critical do people need to be about removing association with anything during the test phase?
That's a great question, and you'll find, I think you'll get, that can be, you'll have different philosophies on that from different labs and certainly in the fear conditioning field. I can say the thing you would really want to look at is your baseline freezing score. So make sure that your protocol has a baseline period in it that allows you to observe their baseline behavior in that context. So what that's going to let you do is see if there's carryover, what's usually called carryover. So if you're moving from one context to another, you'd want to look for very low freezing in the very low carryover, very low freezing in that new context. If you see high freezing, maybe 15, 20% freezing, that might tell you that there's some cues coming through to the animal that you're not able to address, and you would need to take more care changing context if your goal is to really wipe out carryover. Some labs will say, yeah, you need to do it in a different room and use separate doors, it's very isolated. Other labs are able to get successful conditioning with low carryover using the same set of cubicles for training and testing just by altering context, and also paying attention to maybe you have the lights in the lab off when you do testing, but they were on during training. You had the cart that the animals were on covered when you brought them in for testing but not training, or things like that are important things to consider. They're kind of outside the realm of the behavioral tests, but things that need to be considered at any rate.
Those are great thoughts. Thanks, Gerry. All right, well, we've come to our time to close off this session, so a big thank you to everyone for taking time out of their day to attend this webinar. We hope you found the information provided valuable. I'd also like to thank Dr. Gerry Herrera for his time today and his work leading up to this event, and a special thank you to our webinar sponsor, Med Associates. Again, the slide deck and the recording will be available soon for viewing at insidescientific.com. Also, a full transcribed report of our Q&A session will be posted in the coming weeks. So look out for a couple of emails regarding these resources and how you can access them. And also, to second what Gerry said before Q&A, please do take a moment to complete the brief survey that will follow this webinar. Your feedback is extremely important and will help us continue to put together meaningful and educational events. So we thank you in advance. In closing, have a wonderful day, everybody, and we look forward to having you with us again soon at a future Inside Scientific webinar.